Hello, my name is Christine Deal, and I'm going to be covering Chapter 6, Autosomal Inheritance and Disorders. Okay, so I have broken this section down into two smaller videos for ease of viewing. Again, this is just a general highlights of Chapter 6. You are responsible for the reading and highly encouraged to do so. The objectives for this section, just like the other ones, are on your blackboard. I would encourage you to review those, print those, and have those next to you as we go through this section. Okay, so I'd like to start with a quick recap because it's important. I like to do a recap because it helps get your mind in the game, so to speak, and it can be difficult, I know, to just jump right in, especially if it's been a while since viewing the videos or reading. But again, you are encouraged to keep up with your reading because every section builds on the previous section. Okay, so chromosomes, as you recall, are actually large chunks of DNA in a temporary structure containing many genes. And you've got some small chromosomes that have fewer genes, about 80 to 90, and you also have larger chromosomes that have thousands of genes. And so this is important to know because when we get to chromosomal abnormalities, if there is a change within a small chromosome with few genes, then the expression of that is not as great as having a problem with a larger chromosome with more genes. Okay, so a chromosome is with two chromatids is a chunk of DNA that has been replicated during the DNA synthesis S phase of cell division. This structure allows the replicated DNA to be pulled apart during mitosis so that each new daughter cell inherits exactly the right amount of DNA, no more and no less. So the major purpose of packing DNA into the shapes of the 46 chromosomes is to ensure that precise delivery of DNA to the next cell generation. Okay, here's just a visual recap from your previous sections that you've had. Again, I just like this visual. It just talks about, it's just a good viewing of the resting phase and that whole process of replication, and then the G2 phase where there's the growth, preparing for mitosis, and then the four phases of the M phase and the four phases within that of the cell division into two daughter cells. Another visual for a recap of chromosome replication, the interphase, the S phase, the synthesis phase. Okay, and so again, you start with one chromosome that's unduplicated. By the end, you've got a duplicated chromosome, so one chromatid and a sister chromatid that then pull apart and then form those daughter cells with the exact, a very precise amount of DNA within each cell. Here's a little recap again on mitosis. Again, you've got, that metaphase where the chromosomes line up on the equator. Anaphase, they pull apart to separate poles. Then telophase, you've got the nuclei forming. Okay, so you've got two separate nuclei, and then you've got the cytokinesis, where then the cellular split that results in the daughter cells. So again, that's just some refresher visuals for you. Remember, one chromosome of every chromosome pair was inherited from your father, and the other chromosome of the pair was inherited from your mother. The most important issue with chromosomes is that they contain the correct amount of DNA, with nothing extra and nothing missing. So here is a normal human karyotype, where you've got one chromosome from your mom and one chromosome from your dad of each of the numbers. We've got 22 autosomes, and 23 are your sex chromosomes, your X or your Y. Okay, XX or XY. XX for female and XY for male. Okay, so you know what, before we go any further, why is it important for nurses to learn genetics? You may be wondering that. So I just want to take a moment to establish the relevance in nurses knowing all this information. So the National Institutes of Health, they refer to genetics as applicable to the entire spectrum of healthcare, and so therefore certainly, that's the nursing profession, because the majority is a component of that. But then, because the majority of disease risk and health conditions do have a genetic component to them, that of course is influenced by the environment and one's lifestyle, and so that therefore impacts the nursing profession. And that's the whole basis for why we ask about family histories of disease and such. And the top 10 leading causes of mortality in the United States, according to the National Institutes of Health, all have a genetic component, which will be covered in future units. But for example, cardiovascular disease and certainly cancers, there is a high genetic component in that. So it is very important and relevant to understand the concept of the genetics in diseases and human health conditions. Okay, so chromosome shapes were covered in an earlier section, so this is just a recap on that. I'm going to focus on the acrocentric shape. Those would be the ones with a short P arm, which would be, you can see on this picture, numbers 13, 14, 15, 21, and 22. Okay, it's very hard to observe, but it's there. Okay, also the Y chromosome is also has a short P and is considered acrocentric. Okay, so the main thing with the acrocentrics that I want to point out is because they have that short P arm, they have a very they're so small, they carry very little genetic material, so their loss doesn't affect development and normal phenotypic appearance, but the concept of translocation is very important with this because it is the most common translocation, which is the Robertsonian translocation, is more common between acrocentric chromosomes, and that is because the DNA of the centromeres of two acrocentric chromosomes attract each other, whereas the tips of the longer chromosomes have no special attractive force. So let's just page up a little bit. So you can see these long tips, the P arms of the metacentric chromosomes, for example, which would be 1, 1, 3, 16. So they don't have as much attraction to each other as the acrocentric ones do. Okay, so translocation. During gametogenesis, a chromosome can be translocated from its position of a freestanding separate chromosome and attached to another chromosome. And like I've said, that most commonly occurs with those acrocentric chromosomes. When the chromosome material is present in the correct amount, even though its position is different, it is a balanced translocation. Because as long as the genetic material is there, they're going to have a normal phenotype expression. Okay. So a special type of translocation, like I've said, is one between two acrocentric chromosomes. That is known as a Robertsonian translocation. When parts of non-homologous chromosomes are exchanged, but the correct amount of genetic material is present, then the result is a reciprocal translocation. And I've got some pictures of each coming up shortly. So here is, just to focus on the Robertsonian first, you can see you've got two normal chromosomes, and then you can see with this Robertsonian translocation that they fuse together, but there's no loss in genetic material. They're just in a different form, but the genetic material is still there, so that's why it's balanced. Down below is another example. You've got two acrocentric chromosomes. You can see the P arms are very short, and then those become lost, and as I said, the centromeres are very attracting to each other. And so then you've got those two chromosomes combining together at the centromere. So now I've touched a little bit on the first 6 here. About 1 in 900 babies is born with a Robertsonian translocation, which makes it the most common kind of chromosome rearrangement known in people. We've talked about which of the 5 chromosomes are acrocentric. okay? So most of them, a lot of them are balanced. It takes place, the place of two acrocentric chromosomes results in no problems for the person carrying it. So starting at number 7, when we get into the unbalanced forms of a translocation, that's where we start seeing syndromes, okay, and a specific phenotype expression of specific syndromes depending on the chromosomes involved. Okay, and that will be discussed in the next section of part 2 of chapter 6. So here's a picture of a Robertsonian translocation. So you see where you've got, normal non-homologous acrocentric chromosomes, and then they break and rejoin. Small fragment that's frequently lost, and they combine to fuse into one chromosome. But again, all the genetic material is there, so it's still a normal phenotype. Okay, the expression is normal. However, their genotype will be 45 because instead of having 2 separate chromosomes, they now have one fused together. So it'll be 45 instead of 46. But no change in the phenotype. So here's another example. This one is in your book. You've got a normal chromosome 13, a normal chromosome 21. Again, they fuse together, 2 become 1. All the genetic material is still there, so there's no change in the phenotype, but again, the genotype would be a 45 instead of a 46. So a reciprocal translocation also involves 2 chromosomes, but there is a break in each chromosome, and then those 2 chromosomes kind of exchange those broken segments. So you can see how you've got this chromosome 4 in green, chromosome 20 in pink, little sections break off and they kind of switch places with each other. The same genetic material is there, they're just in a little different formation. Again, that would be a normal phenotype as well. And that genotype, because it's still 2 separate chromosomes, it would be a 46. Okay, so that's another difference between the reciprocal versus the Robertsonian translocation. Okay, again, another example of a reciprocal translocation. I love pictures. I think the more visuals, the better, because these can be hard concepts, and so the more you see it, it just makes it easier. So again, little bits exchanged. You can see where that they have exchanged places, broken off and exchanged places. Side-by-side comparison, again, reciprocal, chromosomes break, switch places, and reattach. And then down below, this Robertsonian translocations where you've got 2 that then fuse together to become 1. So again, it makes sense that the reciprocal translocation would be a 46 with the normal phenotype if it's balanced, and then the Robertsonian would be a 45 with the normal phenotype being balanced. Again, another visual, reciprocal versus Robertsonian. Okay. So phenotype effects of translocation. Whenever a translocation is balanced, as I've said, the person's phenotype and function are not affected by the translocation. The gene expression is less affected by differences in location than by differences or variation in the number of alleles or base sequences. Okay, so the location doesn't matter as long as it's all there, basically, is what that means. So potential risk and considerations of these translocations. Problems are common when a person with a balanced translocation reproduces. Okay, so a balanced Robertsonian translocation can result in 3 types of gametes, actually 4 because 1 would be normal, for the affected chromosomes. So you've got gametes with 1 chromosome of a pair, gametes with 2 chromosomes of a pair, gametes with 0 chromosomes of a pair. Possible conceptions are that you have someone who is chromosomally normal, a trisomy, a monosomy, and a balanced translocation carrier. I've got a picture coming up here. Okay, so here we go. This is in your book, so also look there to review it. But here we've got a woman's precursor egg cells with a Robertsonian translocation. So here you've got a translocation with 14 and 21. Okay, in addition, you've got, you've got another 14 and 21. Okay. So possible outcomes of meiosis of this egg would be that you have an ovum with one, because remember that meiosis, they divide and pull apart. So you have an ovum with one separate chromosome 14 and one separate chromosome 21. And then when that egg is fertilized from a male with a normal 14 and a normal 21, then here over here on the far right, that would result in a totally normal genotype and phenotype with two 14s and two 21s. Because remember, you get one from the mom and you get one from the dad. Now, another possible outcome of meiosis of this woman with a balanced translocation would be that she would have an ovum having a balanced translocation with one chromosome 14 and one chromosome 21. So then when that's fertilized with a sperm having a normal 14 and a normal 21, then that becomes a balanced translocation carrier like the mother with two 14s and two 21s. Okay, the third scenario, after meiosis of this woman with a balanced translocation, she could have ovum with an excess of chromosome 14. So you see the chromosome 14 is in pink. So she has two 14s along with one chromosome 21. Now remember, you get one from your mom, one from your dad. So now when that becomes fertilized with a normal karyotype, with normal 14 and a normal 21, then you end up with an extra 14, because here in this scenario, the woman has two 14s, and then you add one from the dad, that makes a trisomy 14. And then you have two chromosome 21s, which is normal. So that would have an expressive phenotype. So in the final scenario here, after this precursor egg with a woman with a Robertsonian translocation, after her egg outcome, possible outcome of meiosis there is that she could have an ovum having an excess of chromosome 21, the ones being in the blue there. She has two 21s and one chromosome 14. So again, when that is fertilized by a male with a normal 14 and a normal 21, that leads to an extra 21 and just two chromosome 14s, which are normal because you get one from your mom, one from your dad. But this extra 21 will result in a Down syndrome individual. Okay, so trisomy, like we've said, involves having an extra copy of one chromosome. So it can result from non-disjunction during gametogenesis, which allows a gamete to retain both chromosomes, okay, as we saw, of a pair, and one gamete to have no chromosome for that pair. Non-disjunction is more common with advancing maternal age, but it can also result from gamete formation in a person who is a balanced translocation carrier, which is what we just viewed over here on this far right. Okay. But most commonly, trisomies occur from a non-disjunction during gametogenesis. And so here's a recap on non-disjunction. Remember in metaphase, everything, they all line up on the equator there, and then they pull apart. They should pull apart equally with an equal number on both sides. In this case, that did not happen, and so certainly you're going to have a difference there in the genetic material, or the genetic number, chromosomal number. So here's another picture of non-disjunction. So you certainly end up with some daughter cells here that have less chromosomes and some that have more chromosomes when they don't split apart evenly. Okay, and I think that wraps up part 1. Okay, so part 2, we will talk about more of the syndromes, okay, associated with trisomies, and we'll go from there. Okay, thank you. If you have any questions, please email me, call me. My number and my email are all on Blackboard. I'd be happy to help. So if you have any questions, please feel free to contact me. I'm happy to do so. Happy to help. Thank you so much.